She's a hard act to follow. So I was trained as an MD at Stanford. I arrived from Montana in the fall of 1960. Stanford was the only medical school I applied to because they were the only one that developed a curriculum that would allow you to do medical school in 5 years instead of 4. You may think that's not an advantage, but it meant that you had a half time every day for the first 3.5 years to do research while you were learning medicine. That was important for me and important for everybody who went through that program, because by the time we graduated, we knew what we were going to do. And we knew not from word of mouth, but we knew because we had entered this realm of advancing medicine through research. So now I'll tell you about some of the research. Many years ago, following the radiation that killed people in Hiroshima and Nagasaki, a lot of scientists wondered what the radiation was doing. And what it was doing at the lowest dose that kills was wiping out the blood-forming system. A lot of research was done, but we followed that up by showing that not only was the bone marrow, here and here, everywhere, the source of blood formation, but within the bone marrow, can you see? Okay. There was a population of cells, which we called stem cells. Within their ability to make all of the blood cells, these cells are the only ones when they divide to give rise to 2 daughter cells, at least one is still a stem cell. So the very next one down, as I'll show you in yet another slide, cannot self-renew. So it has a limited lifespan. It divides a lot of times, maybe 20 times to go from a single cell to 1 million daughter cells. And then the next one, 1 million daughter cells. So we knew that these had to be important, but we didn't know if we could harness their function to replace bone marrow transplants. And let me tell you, all of the bone marrow transplanters said it was a waste of time. I learned, and I'll say these kinds of things over and over again, that the people who'd gone through 4-year medical schools where you had to be an ace memorizer even to get into medical school, and then to beat out the others for the best post-medical training, you had to memorize more. And so I was talking to a bunch of people who are great clinicians because they've memorized everything they need to know, and they can put together signs and symptoms to treat a disease, but they know nothing about changing the paradigm through research. So I've learned over the years always to smile when I'm talking to my clinician friends, but not to listen to it. It'll hit me. So because this property is self-renewal, it only becomes important to ask, well, is it just blood that has self-renewing stem cells? And can blood stem cells turn into brain or liver? And the answer is no. It only makes blood. Some bird says, oh yeah, and only blood. Because they are the only self-renewing cells, you should think about them in the development of the blood-forming system, maintaining the blood-forming system, and this is more and more, although I'm going to change that a little bit, in finding out what goes wrong if you have a blood disease. And I would argue right now, any blood disease that you have, other than one that you took the wrong drug and it wiped out all of your blood-forming tissue, or you had an antibody response against your own platelets or red cells, except for those, I'm going to argue first you should look for a derangement of stem cells. It's not perfectly clear from what I'm saying anything yet, but hopefully. What I want to end up having you understand is that for the next generation, it's hard for this generation, this is the platform for regenerative medicine. So how could they treat chronic diseases and cancers that are currently incurable? Well, because they're the only cells that make more of themselves, you should purify those cells to see what changes they've made if things go wrong. Because they make more of themselves, if you transplant them into an individual who you've prepared so that the cells you put in will take the place of the dead cells that were just killed by chemotherapy or radiation, these are single treatments for life. So if you had a genetic disease, say sickle cell anemia, your own stem cells, every one of your blood stem cells has the genetic abnormality. But if you replace it with your sibling or your mother or your child's blood-forming stem cells, if they don't have the gene that causes sickle cell anemia, then they would go in, they would replace the old stem cells, and for life, because they self-renew, you don't need another treatment.
Wow.
This is partly why the pharmaceutical companies don't like us either. Because it's once for life. Okay, there are curative therapies for blood diseases, but there's a whole bunch of areas in transplant that I'll show you that I think will be facilitated by this platform of blood-forming stem cells. Just go to the next one, because everything else I'll mention if I spend too much time. So, one of my former students made this slide, just to show you, for the blood-forming system alone, that blood-forming stem cell that makes blood, these are all of the diseases that we have tested in the mouse models of the disease, are tested with the actual human cells that we believe we can cure with a single transplant of a healthy blood-forming stem cell. For those who can't read, sickle cell, beta thalassemia, Fanconi anemia, aplastic anemia, and so a whole bunch of diseases. So the promise is there if we can convince the memorizers that it's worth doing. Over the last 30 years, my lab isolated not only the blood-forming stem cells, so now we're going to go down instead of across. Its daughter cell that is not a stem cell, at the single cell level, can make all of the blood cells. But because it doesn't self-renew, it only does it for about 8 weeks. So that means if you're going to have a continuous rescue from a radiation accident, like Hiroshima and Nagasaki, or therapy for sickle cell or other diseases, this is the only important cell. Okay? This is, you remember the Gerald Ford thing? Can't chew gum and walk at the same time? All right, so now we want to know where the blood-forming stem cells are. We proposed a long time ago that there would be a little home for them that would nurture the stem cells. And then as they divided and gave rise to the non-stem cell, they'd all go into different regions of the bone marrow. And I'll just say this was not universally accepted. Just 2 years ago, 30 years after we had isolated the stem cell first, we found a way to see where they are in the bone marrow. Now with these lights, you probably can't see anything, but I'll just describe what you should imagine you're seeing. So we have mice in which the true blood-forming stem cells, and only the blood-forming stem cells, make a red fluorescent protein. So if you look through a microscope, see that? Can you see that as red? And every time there's an arrow, that's the cheater, the arrow. And what I have in green was the blood vessels in the bone marrow. Now, before we published this experiment, the experts, or at least the self-proclaimed experts, at the Stanford of the East in Boston, Mass, claimed that the home for stem cells was right next to the bone. And they developed experiments, clinical trials, none of which worked. But we found, this is observation, not hypothesis, we found that they were stuck to the outside of the blood vessel that has most of the traffic between the bone marrow, the blood-forming organ, and the rest of the body. And within a mouse's femur, there are just 100 cells. And if you look at this, there's not even 2 cells next to each other that are red. So if there is a daughter cell that is still a stem cell, it must escape and go somewhere else. And we found in an odd experiment, which we call parabiosis, that if we have 2 mice whose blood circulations are connected together, stem cells from one once in a while go into the blood and go into the blood-forming organ of the other. So they know how to go out of the bone marrow into the blood, and lucky for all of us and the bone marrow transplanters, they know how to get from the blood back to the bone marrow. It's the only stem cell that can go from the blood to the tissue to regenerate the tissue. I assure you, when we injected brain stem cells into the blood of a mouse, they didn't go back to the brain. So it's a peculiar and evolved property of these cells in all of the vertebrates at least, and as these two will tell you, even in the link between vertebrates and invertebrates, you have a blood-forming stem cell population. And it's developed these properties to move from one part of the body to the next, and luckily it has the information so the first bone marrow transplant could work. So if we wanted to do our studies on stem cells and locate them, we had a way to separate them. And this goes back to a story I'm not going to say in full, just to say that at Stanford, engineers and physicists were brought together by Joshua Lederberg, the head of the genetics department, with smart, then young, now not so young, or not necessarily alive, people who wanted to develop a machine to sort cells. And the Herzenbergs did develop such a machine. Would not have happened without that physicists, early computer scientists, engineers, working with biologists. Would not have happened. And so I would doubt even today all of the advances that came from it would be there. So now we're going to ask, of all those cells, 1 in 100,000, so I've overrepresented it, is a blood-forming stem cell. And we want pure blood-forming stem cells so we can understand what they do. So Herzenbergs invented a cell sorter which we used with a number of reagents to purify stem cells away from cancer cells, other cells, and as I'll tell you, the most important cells that cause rejection of tissue and organs, T cells. I co-founded a company called Systemics because then, and probably now, but then, the university didn't want us to do the application of the discovery for clinical therapy. They were very risk averse, I'll just say that. And since we had developed a method not only to isolate stem cells, but to make a mouse that had a human blood-forming immune system, and Mike McCune and I were putting HIV at the beginning of the AIDS epidemic in them to see how HIV works, the lawyer said, not at our place. So we co-founded the company. It wasn't commercially free. At least those of us who found it. Everybody else who went there, you could say maybe something different. So one of the therapies that we wanted to test is, could you give a woman who had metastatic breast cancer, so it spread beyond the breast, spread beyond the lymph nodes, but in the bones, in the liver, in the lungs, so there's no way surgery or radiotherapy could cure it. You have to give high-dose chemotherapy, and you have to use combinations because each drug has a side effect, and the side effects are as bad as killing the cancer cell. So if you have 4 or 5 different drugs that all focus on the cancer cell but have different side effects, you can get to the point where you say, am I close enough that I might cure this disease by killing all the cancer cells in the body? So these women had their blood-forming tissue removed, stored frozen, given a lethal dose of combination chemotherapy, thawed the cells, reintroduced them, and let me tell you, that first patient, when I saw her getting a thimbleful of what looked like clear water, and then 10 days later, her white cells were back to normal. It was an amazing, amazing thing, but I was scared. So, when we discovered stem cells, this is another anecdote. When we discovered stem cells, everybody who did a bone marrow transplant called themselves bone marrow transplanters, and the titles of the paper were bone marrow transplantation. After we discovered stem cells, without changing anything they did or do, they said, we're doing stem cell transplants. So there's a whole bunch of people walking around thinking they got a stem cell transplant, but they didn't. Now here's why it's important to be accurate about what you say you're doing. When we looked at the bone marrow, or what's called the mobilized blood, it's not important, from women with metastatic breast cancer, we found the cancer cells in the blood. So why would you go through putting the woman through hell, next to death, and then inject cancer cells back in, right? I said, this is obvious, you don't want to do that. Breast cancer oncologist, bone marrow transplanters said, why do you think if you put the cells back in, they'll grow? I said, it's a century of cancer biology that we have done. Haven't you read any of it? So they were against doing the trial. Nevertheless, we did the trial. So this is the survival of women who got unpurified mobilized blood to rescue them from their high-dose combination chemotherapy. Half of them were dead in 2 years. All of them are dead now. It's 20 years. That's no better than the drugs they give then and now. But here's the cohort of women who got back pure stem cells, which we showed were at least 250,000-fold depleted of the breast cancer cells that were in there at the start. And now they're gone. So the only thing we saved them with were cancer-free, blood-forming stem cells. Half of them dead, 10 years. Now, 22 years from the first transplant, 1/3 are alive, still without disease. When you go to your doctor, you could ask your breast cancer oncologist, can I have that therapy? And the answer is no. And it's no for another reason that none of us suspect. Systemics, the company I co-founded, made a partnership with a very large Swiss pharmaceutical firm who said, our interest is the same as yours, and we have billions of dollars to take it to the people faster. That lasted for 4 years, until they found a drug you could take by mouth. Didn't treat these diseases, but made several $1 billion a year. So they concentrated their business assets On the other thing, they didn't call me up and said we're blocking it. They just dropped it off. I'll just say that we have it back now at Stanford in a not-for-profit setting. But it was one of the many times I realized that although we have a model that you should make discoveries, whether it's in industry or academia, that it's industry that really knows how to make cures. It's wrong. Industry has a clear and required goal to make a profit. The function of a company is to make a profit. If you try to say to the directors or the shareholders, let's not do that, let's do something that might, if it worked, save more people, you won't get much. You know that, you may not believe it, but that's a fact. So this is not a therapy. Even though we did it in 96 to 98 and analyzed and published in 2012, just now we raised the money so we can do it in a not-for-profit setting. Because if we save 1/3 of the women, the 50,000 women a year in the US who get metastatic breast cancer stage, that's more than 15,000 a year, and we could have been doing it since 2000. So this is not just a little philosophical debate. This is the reality. So I'm going to come back to this at the end, and it might be a few hours later at the rate I'm going, but we have to deal with this reality or we will not change stem cell or any other therapy. I'm not a communist. I know at some point you have to have a commercial entity to distribute it. But I don't want to give it to them too early because they make mistakes and they have other things in their docket and they can make a choice for the one that makes more money faster. I don't know how many people I've pissed off, but it should be a lot. Now, we got the stem cell. I obtained, thanks to 2 postdocs from Japan, Toshi Miyamoto, Koichi Akashi, bone marrow that had been saved from people in a Hiroshima hospital who got acute leukemia after the bomb. They had saved it as living cells frozen down. So we could ask the question, of all those cells that I showed you in that chart, is there a cell that really is the leukemia cell? And so we sorted the cells by the kinds of markers we have, and we knew these had the markers that were blood-forming LTs for long-term stem cells, okay? And we could get them pure. And then the very next cell down, short-term stem cell or multipotent progenitor, we showed was the leukemia stem cell. We transplanted it from patients into mice that have no immune system. This cell doesn't transfer leukemia. These always transfer leukemia. Their daughter cells lose the ability to transfer the leukemia. So the leukemia stem cell, not a normal stem cell, not even the same marker as a normal stem cell, but a cell that can self-renew on its own and expand in limitless fashion, is this cell. So now we can start to look at this cell and this cell. And what we found is that particular chromosomal change that had occurred in these patients and was exactly the same in every leukemia stem cell in the body was also present in normal stem cells, anywhere from 1% to 40% depending on the patient. And it had that change in the chromosome. But when we put it in a tissue culture tube, when we transferred it to immune deficient mice, it only transferred normal blood formation. So that was the first big clue that between here and here, more has to happen to give rise to leukemia. Okay? We saw this in every kind of leukemia, whether it's chronic myelogenous leukemia, acute leukemia and acute leukemia with this genetic or that genetic predisposition. It was always almost the same. So we raised a hypothesis first in 1998 and 2001, where we said mutations can happen in any cell in the body. And the mutations from whatever it is, gamma ray, too much oxygen-free radicals in your food or whatever, it happens randomly in any cell, and these are only 1 in 100,000 cells. But if a mutation happened in this cell that's going down here, it only has a limited lifespan, it's gone with the lifespan of that cell. These are the names of the kinds of mutations that we saw in mouse leukemia and in human leukemia. So we proposed way back then that the leukemia stem cell, let's say it had all 7 of these events, had to come from a cell that had 6 events, and that from a cell that had 5. The reason that we thought that had to be true is randomly, no single cell is going to have 2 mutations at once, both of which drive the formation of the leukemia from the normal cell. Okay, so it has to happen one at a time. So we raised the hypothesis and tested it, and I'll show you proof that in fact it's the unlucky roll of the dice if the mutation is in your, and maybe only 1 in 100 of those will drive leukemia. We live a long time, and as we get older, we tend to get myelogenous leukemia. So we wanted to see if that hypothesis is correct, so that if this is 7, we ought to find a cell that has just the first 6, and another one that has just the first 5 mutations, all the way up to one that has none or 1 mutation. So that's the experiment we devised. So there's a whole bunch of medical jargon here. Simply to say, we took the acute myelogenous leukemia cell, the leukemia stem cell, and did a partial genome sequence. So we could see where a normal cell, like a T cell in the body, will give us the baseline, and then the leukemia cell will have changes. Here's one change, here's another, here's another, here's another. These are all the changes, and these are the actual proteins that will change because of a mutation that changed their sequence. The ones that are in red come up in many, many leukemias. The others don't. Mutations are random. So for the most part, they aren't going to hit a gene that will cause something to happen. And what happens is that that little home for blood-forming stem cells is a site of competition with stem cells that grow better. So you can see here in the drawing and in fact, all of those changes, let's say a cell that has 1 + 2, that expands, but it doesn't expand all of the cells that have the markers of stem cells, because it expands, competes for those niches, so it expands at the expense of the normal stem cells. This is the most important concept I can try to get across. A stem cell or a mutant stem cell, if it has a way to outcompete a normal stem cell, will eventually take over. These are the names of the genes. So we tested by making a DNA test that was exquisitely sensitive, that would see only the mutant form, not the normal form. So we made what's called DNA primers for all of those, and then we took single blood-forming stem cells from that patient's bone marrow. And we asked, did it have all the mutations, or less than all, or 1 or none? Because you could do a single cell test. And this is the result of the first 5 patients. I'll show you 3. There's no question that patient number 70, the one you saw, the very first mutation led to a group of cells coming from that single cell, and whenever you have a group of cells coming from a single cell, we call it a clone. The clone is expanding. It's still in the bone marrow. It has a mutation in a known gene. All of us have 2 copies of any of our known genes on our 2 chromosomes, one from our father, one from the mother. And in these 2 first patients, the second mutation that was productive in expanding that cell type was the same gene, and it was a loss of function. So that cell had some important aspect of regulating cell numbers, that gene. And there were other genes, and we finally got to the end. And the main point here is that in every leukemia that we looked at, now it's over 30, the early mutations are in genes that regulate the expression of other genes. And only at the end do you get the famous cancer genes that expand the population. So that's what I usually say because I have a title, but I'll just say it to you. If you want to study what causes human cancer, don't ask a scientist who studies oncogenes, ask the cancer. Just go get the cancer, get the answer, and then you know you're on the right track because it caused the cancer. Okay, so in acute myelodysplastic leukemia, we can say every one of the patients, the progression is in a blood stem cell clone, which competes with the normal cells. There are no leukemias we've ever found of the stem cell itself. So that home next to the blood vessel somehow regulates the number absolutely. Every tissue in our body that's been examined carefully has its own tissue-specific stem cell. Brains have brain stem cells. Muscle has muscle stem cells. Bone and cartilage have a common bone cartilage stem cell. Liver, it just turns out, actually has a stem cell. So now we have the same kind of hierarchy of stem cell and non-stem cell donors. So we propose, and we're going to test, that this will happen in all cancers. And we're testing. I think we're the only ones in the world who are testing because we're the only ones who believed our own papers. And we propose then, and now we know, that if that first mutation that allows one stem cell to outcompete another is aberrant in some way, it could cause a disease if and when it becomes the dominant stem cell. So if you know anybody with Myelodysplastic Syndrome, a disease of people my age, you don't have enough red cells, you don't have enough platelets, you don't have enough neutrophils, so you're infected or bleed or anemic. Myelodysplastic syndrome, Wendy Pang in my lab showed, absolutely starts with a single cell that takes over all the definable niches we could see, at least 98%. So whatever its genetic abnormality, it is very competitive at the level of the stem cell, but it can't make blood normally. So that's why I said one should rethink every blood disease, because you would have thought this is going to be a disease of the cells that make red cells if your patient comes in with anemia. But it's at the stem cell level. I'll skip this one. Now, we had proposed and we showed, as I told you in the leukemia, that there would be cancer stem cells and they would accumulate during the run-up to the cancer, those mutations. When they are a cancer stem cell, they're still trying to make some kind of normal offspring. Cancer stem cells, whether it's the cancer stem cell in the breast for breast cancer, or the cancer stem cell for leukemia in the bone marrow, make up about 5% of the cells. If you don't purify the cancer stem cells, you'll be looking at red herrings all over the place of the genes or the properties of all those cells that are not. But that hypothesis led Sean Morrison, Tanisha DeRay, Mike Clark, and I to propose that if you don't get rid of the leukemia stem cells in the whole cancer, the whole leukemia, it'll regrow. But if you find a target that's on all of the leukemia stem cells, you've got a chance to eliminate the cancer. So we began looking at the difference between the purified leukemia stem cells and the normal blood-forming stem cells or multipotent progenitors. You could do that, and you do a side-by-side test, and you get something. We found the name CD47 of a gene that was on at very high levels in the leukemia, very low levels in the normal stem cells. And we found that it was on the surface of a cell. And by our work and actually reading some other people's papers, we found it's a don't eat me signal for the scavenger macrophages in the body. So we have scavenger cells that are like amoeba, that crawl around the body, grab a hold somehow of a cell that's dangerous or dying or inflamed or infected, and eat it. But these cells, this molecule, CD47, is a don't eat me signal. So here comes the macrophage. It's binding. It sees an eat me signal, but there is also a dominant don't eat me signal, so it can't eat it. This is actually how it works. There we go. We made an antibody that blocked the don't eat me signal. Now the cell comes in. It doesn't get the don't eat me signal, the macrophage. So it eats it. And if you think that's all just cartoons, here's a leukemia where we block the don't eat me signal. It's all inside the eating cell and didn't block it. They're outside the eating cell. Then we found that the don't eat me signal is on all human cancers. My colleague Mike Clark found the breast cancer stem cell. He's at Stanford. He transplanted them into the breast of immune deficient mice. They have no immune system themselves. They have macrophages, but they don't have the immune system that could reject it as a graft. We let it grow for many weeks, and then we treated them with a blocking antibody or another antibody that doesn't have the same specificity. And as you can see here, there's no cancer. That works pretty well. Same for a brain cancer of children, medulloblastoma. Same for glioblastoma, teenagers and adults. That we could treat them with a human tumor in the mouse's body with antibodies that block the don't eat me signal. And if we start with small enough tumors or small enough numbers of cells, we can clear all of those cancers. Now, that's the good news. The bad news is none of us show up with that tiny amount of cancer in our body at diagnosis. So you've got to have some way to debulk the tumor to make this therapy work. It's in late phase 1 clinical trials. There are good results in some I'll show you, but I'll say that this led us, after funding entirely by the state from the California Institute of Regenerative Medicine, to go from our discovery of the molecule, the discovery of the antibody that blocks it, testing that it does block these human cancers, then changing the antibody so it could be used as a therapeutic in humans, testing in the right animal models that we could deliver it in a certain way without toxicity, getting together with people who know how to write things for the FDA to get an initial new drug application, and then get approved to do phase 1 trial. That alone usually takes 15 years, 10 to 15 years. We did it at the university with no professionals in this field. And I think we did it from state funding. If you bought some of the bonds for CIRM, you know that you're part of this. And so without commercial intent, we could look for advancing medical science. Now, we did find that our antibody synergizes on the cancer cell surface, like a lymphoma, with other therapeutic antibodies, rituximab for lymphoma, Herceptin for certain kinds of breast cancer, cetuximab for colorectal cancer and pancreatic cancer, and we published that science. These, when we tested in a clinical test, or previous almost clinical test, this is the growth of the non-Hodgkin's lymphoma in mice that are immune deficient. We give our antibody, it stops it dead, but it didn't get rid of all of them. Rituximab, about the same, but together they got rid of all of them. So it's been reported in a clinical trial, early days, that half of the people with what's called relapsed and refractory large cell lymphoma or follicular lymphoma respond, and 80% of them, now over about a year and a half, have no measurable disease. So the science we did, the preclinical testing we did, gets us at least the first lead of a cancer that we believe we'll be able to treat. And of course, because I'm a co-founder of 47A and a director and have stock, you might think I'm biased. But meanwhile, in the lab, with really terrific fellows from around the world, we found that in the arteries of humans, the atherosclerosis that develops has CD47 on it. The cells that are proliferating to close off the coronaries, close off the carotids for a stroke. Destroy the aorta so it balloons out in an aneurysm. Nick Leeper did that, and Yoko Kajima. Berlinda Vernig and I did it for an over-proliferation of a cell called a fibroblast that normally is part of a scar that heals tissues, but these keep proliferating and they cause scleroderma, or pulmonary fibrosis, or NASH, non-alcoholic steatotic hepatitis, and you die with those diseases. My son, my oldest son, had NASH and had to have a liver transplant. He's alive, but I'm just saying that these are serious and not uncommon diseases. And their commonality right now is CD47, and we are testing the preclinical. In the mouse, the model disease, you can handle it with the anti-CD47 antibody. Now, I know time is marching on, but I'll try to finish this last part, because I want to get back to the platform for regenerative medicine. As Kay mentioned early on, at a company called Stem Cells Inc., Nobuko Uchida, my former student, and my wife, Anne, Tsukamoto Weissman or Weissman Tsukamoto, I can't remember which way it goes, isolated the human brain-forming stem cells, found a way to grow it up to millions of cells that are still brain-forming stem cells. And I have to say from the beginning, this is a brain-forming stem cells, and we don't know yet if the different parts of the brain have different brain-forming stem cells. But when we transplanted them into a mouse that had a spinal cord injury and was paralyzed, the brown spots are the daughters of the human brain-forming stem cells rushing in. This is dead cell zone. This is toward the head, that's toward the tail. And you see there's lots and lots of brown things. Those are the human daughters of the brain-forming stem cells, which are rushing in to replace cells that have been killed because they didn't have enough oxygen or food supply because the blood supply was interrupted by the spinal cord injury, just like Christopher Reeve's spinal cord injury. It wasn't a cut. I could have been using that. It wasn't a cut. It's a loss of blood supply. So this is a mouse that has the disease and got what's called mesenchymal cells. And what you can see is he drags his left feet, he's not coordinated, the tail is not acting in a coordinated fashion. This is a mouse, same lesion, same disease, injected 8 weeks before, above and below the spinal cord, with human brain stem cells, and the animal's cured. In that process, we found that we could treat a large number of diseases in early phase clinical trials. But we also found out that it's hard to raise money for that. And that this is one of those things. When people who have the money or are hired by the money take over the leadership, after a while they actually believe they understand what you're doing. And then they make business decisions on the basis of what they understand. This company went broke because of one of those decisions, and we're bringing it back both at Stanford and maybe in China. But simply to say, we did treat tens of patients with thoracic spinal cord injury, and half of them regained sensation. We treated people with macular degeneration, and the eye that got these cells didn't progress, whereas the other eye continued to progress. We treated about 8 kids with genetic diseases, congenital genetic disease, the brain-forming system. And it looks like it was working. It will come back, but all of this will get to my final point if I ever get there. Now, let's say that one of you has a blood-forming system, and even if you're my brother or sister, it needs to be replaced. So you're going to use my bone marrow. My bone marrow, unfortunately, has T cells in it, the immune cells. So T cells go from the blood-forming stem cell giving rise to a precursor of a T cell that goes up to an organ called the thymus. That's why it's called T for thymus. And then they go out and they reject anybody else's skin, brother or sister, or heart, or lung. So we knew that T cells could be dangerous. And in fact, when you transplant bone marrow that has T cells in it, every system of the body, this is the hands of the people, has an immune reaction of the donor T cells against the recipient's tissues. And although 28 years ago we showed that we could do pure stem cells that have no T cells in it, they grow up and they accept, while they grow up in the thymus, they accept the body as self. So they delete the T cells that would have caused this immune attack. And there's no immune attack. We've proved that long ago. Then we showed what I actually started doing in high school in Great Falls, Montana, to ask whether a bone marrow transplant, which gives rise to an immune system of the donor growing up in the host, if that would reject an organ from the blood-forming cell donor or the stem cell donor. And the answer is it doesn't. So now if you have an organ graft, you get immunosuppression for life. There, it's one-time transfer. You can put the heart on or the other organ in, that's the liver, at the same time or a year later, and it will never be rejected. It's an important precedent because here we're looking at mice that get juvenile diabetes because they have the genetics like people do, That's early onset juvenile diabetes. Juvenile diabetes is a disease in which your T cells that leave the thymus did not get rid of the ones that were anti-self. So I have some, for example, that'll see part of the insulin protein is foreign, and they'll go into the pancreatic islets and they'll kill them, and then we get diabetes. Okay, so these are mice with the same thing. We prepared these mice for transplant with irradiation, some antibodies, and we gave them back their own bone marrow. It bought them a month. All we did is we got rid of some of the T cells with all the radiation, but they came back. Even if we did stem cells, no T cells there in the graft, another month. But if we transplanted the stem cells from a donor that was genetically resistant to get the autoimmune disease, that is, it could censor the developing T cells in the thymus so the ones that are against self don't emerge, don't go to the pancreas, don't cause the disease, we cure it. And if you have some that were diabetic so long, curing the autoimmune part of the disease doesn't cure the disease, we co-transplanted the insulin-producing cells and the blood-forming stem cells from the same donor, and it worked. Published that 18 years ago. Nobody's been treated that way. Now, if you want to treat a kid who is peeing and drinking too much, 5, 6, 7, 10-year-old, the last thing you want to do is take them next to death with radiation and chemo in order to get the stem cell graft in, and since bone marrow transplanters don't use pure stem cells, they'll get GVH. We knew that we had a barrier, so my students and I began looking for how can we get rid of the cells we need to get rid of and still get the transplant to take. So Aniska Chekovich, Gita Bhattacharya, and I said, let's take antibodies out that we made that only bind to blood-forming stem cells. And we found one that eliminated blood-forming stem cells. And we had another 2 to get rid of T cells. So now we did exactly the same that radiation did. We got rid of the T cells, we got rid of the diseased blood-forming stem cells, and to the point that we got the graft accepted. So what we wanted was to get to limited or no use of radiation or cytotoxic drugs, so that we could do sickle cell, so that we could do a particular kind of anemia called thalassemia A, mainly in Korean populations, and because many of them are Catholic, they refuse to have an abortion even if they know, and the child dies late gestation or just after birth. So you would even think of doing this before the child's born, but not if you need radiation and chemo that will stop the growth of the brain, the bones, and so on. So we remove the immune reactive cells and the disease stem cells within it. And as I'll show you in a minute, if we co-transplant an organ with the stem cells from the donor of the organ, we get it to succeed. So we found a way to make the antibodies against stem cells and T cells better. I'm not going to go over it. But here's an unpublished study from an MD PhD student in the lab, Benson George. We transplanted from a particular donor stem cells, into antibody conditioned hosts. And then from that donor, we transplanted newborn mouse heart, because if we could get it in, it would survive the transplant. The ones that were not conditioned with the antibodies did not get the right stem cell transplant, rejected it within 10 days. Those that matched the stem cell and the heart, that was out 100 days, it's now 300 days. So all of this sounds like science fiction, but it's all moving toward the clinic. So these are the diseases. I'm not going to go over it. But for us to transplant a liver or a heart or a lung, somebody has to die. So either your liver, heart, or lung, you had to have the motorcycle accident or the gunshot to the head. When my son had a liver transplant, I took him to the place that had a lot of people shot and dying in accidents, New Orleans. And at the Ochsner Hospital, where they do 300 liver transplants a year, to miss 300 donors. And they did the logistics to get it in. He was saved. But not everybody has the time or the resources or the will or the knowledge to find a place where there's a donor. There's hardly any donors here. The last place you would get a liver transplant is right here, even though we have the best people to do it. Just don't have enough donors. But we wanted to be able to have cells in a dish that came from somebody that's known, that would make blood forming and, let's say, heart forming, or blood forming and brain forming stem cells. We did it. A young assistant professor, Kyle Lowe, has taken human embryonic stem cells, those that are not allowed to be the object of research in Governor Pence's Indiana. Oh, ex-Governor Pence. Not allowed, criminal, to do it. But we made embryonic stem cells into bone and cartilage, smooth muscle, heart, and now we're almost there, blood-forming stem cells. So we can go to a dish and say, okay, what is the match? Of all the embryonic stem cell lines we have, do we have a match for you? And then we'll pull them out, we'll do antibody conditioning, and hopefully get there. Now my very last point is this. I've been alluding to it, and I just want to try to clarify it for everybody. Right now, discoveries are made in academic institutions. Large drug companies are no longer in the discovery business. So usually, in an academic enterprise, you make a discovery, then you go out and sell it to a company or form a company. If you do it early, and it takes 10 years even to know if it's going to work, that's called the valley of death. Why is it a valley of death? Because you need more and more money, and the money makes a decision whether to keep spending money on it in order to get the possible return, and risk comes in, and the smartest person in any company or any venture capital group will say it won't work, and thereby stop the flow of money. It is the valley of death. We need to be able to be here. So that's why, with Bob Klein and a group of people in California, in 2002 to 2004, we wrote Proposition 71. The churches and the anti-groups held it up from 2004 to 2007 before, after a high court decision, we were allowed to spend the money in California. And we made a number of specifications of what the agency should be like. The one I just want to tell you of is that the NIH supports this. They say they support that, but they don't. If they tell you they do, I can tell you they're not serious. But CIRM, California Institute of Regenerative Medicine, has the right to look at discoveries, and if the discoverer applies to CIRM and says, I want to build a team, and I want to do that translation and clinical trials at an academic institution without commercial input and therefore commercial obligations, you can have it. Now, from 2007 till now, we've had about 10 or 11 years. You know that it takes 10 to 15 years to get there. But there are over 30 clinical trials funded by CIRM, including the one I mentioned about anti-CD47 and lymphoma. I weigh in that if you take money from the state, and you're at a university, the university has to share the royalties with that state agency. It's not allowed for federal funding. It's called the Bayh-Dole Act. The universities don't have to give a penny back to the ones that funded their research. So there's a whole bunch of experiments we're doing here in how you understand and how you advance medical discoveries to clinical translation. And I've worn myself out, so I probably worn you out. Thank you. So if people want to ask questions, Benjamin, Dr. Rosenthal will bring the microphone.
Hi, thank you so much. This was incredibly enlightening. I was wondering if you could talk a little bit about induced pluripotent stem cells and in the future you think those will play in your research as embryonic stem cells are controversial.
Perfect. So here's what happens. Embryonic stem cells, so-called embryonic stem cells, are derived from unused fertilized eggs in vitro fertilization clinics. All of the embryonic cell lines that come from that. That does not represent all races and economic classes. But thanks to Shinya Yamanaka and the people who preceded him in scientific discoveries, we can convert, we meaning everybody, not me personally, can convert a skin cell or a blood cell to the same stage as the embryonic stem cell, backwards. You have to put in several genes, and they are cancer-causing genes, so you got to control them. But Kyle Lowe has gone through, and for those last slide I showed you, the development of heart cells or bone cells, it works exactly the same with IPS cells. So this is way more democratic. It means that you can build up banks of cells that will fit any ethnic group, any economic group, and so on.
So I have a question regarding ulcerative colitis, and I was wondering, we're not talking about replacing the organ, but is there something like this going on, and in what time frame is that actually currently available clinically?
Yeah, actually, a discovery that we made in the 1980s showed that the immune cells that cause ulcerative colitis or Crohn's disease, gastrointestinal tract, immune inflammatory diseases use a particular homing receptor to go there. And a company, without paying us anything, made such an antibody in humans, that's vedolizumab, and it is having an effect on that disease. It's in the late part. Insofar as your immune cells that attack yourself, and that's what ulcerative colitis sort of is, has a genetic component that it's doing that abnormally, eventually we can use all antibody conditioning, outpatient, and stem cells from a family member who does not have the same genetic predilection. Now that's not going to happen tomorrow or even 5 years from now, but it is going to happen.
I had a question. Do any of these single treatment therapies, once in a lifetime therapies, reach the commercial stage or are available outside an academic setting?
Say it again.
Say it again. Have any of these single once in a lifetime treatments that you mentioned, you treat them once in life, reached any kind of non-academic setting or commercial setting? Are they available to the general population?
I don't think it'll be spread widely enough that it gets to the general public unless it's commercialized. So I know that you have to go down the road as far as you can so that it can be spread to hospitals and so on. So I believe that it will come. Of course, the venture capital and the big pharma who invested in it early said, well, let's see, how much does it take to separate those cells? Well, it's about 12,000 back when we first did that cancer treatment. And we asked back at that time for women with breast cancer, well, could we charge $25,000 so the company could make some money? And the breast cancer doctor said, we're not going to give it up. And they thought it was way too much. And then after that came these therapies. And you and I know there are therapies now that are not one time that cost you $85,000 to $150,000 over a lifespan. And some of the insurance companies do pay. But some countries don't have insurance companies that pay, so they're not available to everyone. So I think there's a lot of being at the pushing the envelope about not only healthcare and the advancement of science for healthcare, but how do you make sure everybody gets it, and if those can't afford it, get it at a lower price. What we did in Proposition 71 is we said, if you as a company license this, you not only have to give your royalties back to the university and whoever else was involved in it, but you have to give near your costs, reasonably presented, for those people who can't afford the treatment. So it's Californians only. It's never been tested because it takes a long time to go from a discovery to a cure. But that gets at that issue.
I have a question about the CD47. So, do you know what the function of that is in the body? Because if you disable it, well, does that mean that there's some part of the body now that normally needs it to stay protected that is now being attacked, meaning there could be side effects?
Absolutely. So we found a way to deliver the antibodies, and with people in a first world country in a clean environment, we don't put them at risk. There's no toxicity. But if you kept getting those antibodies for life, I'm pretty sure you'd start getting some toxicities. But we're early enough in the clinical trials, we don't know what happens, and especially when you go to a third world country and try the same therapies, where they are, like North Carolina, in high danger of infectious consequences.
What is your opinion on using placenta stem cells for regeneration of cartilage and knees? I'm 4 months into it.
Say the last part, the disease?
Actually, regeneration of cartilage in your knees.
So, I haven't seen the evidence. The placenta has blood-forming stem cells that we showed and HanaHaus showed makes blood and only blood. Somewhere in the placenta and in the bone marrow there are mesenchymal cells, a tiny fraction of which are stem cells. Charles Chan, Mike Longacre, and I showed they make bone and cartilage and nothing else. So I would be skeptical. And I want to actually, since everybody here has the chance, if you Google stem cell and therapy, the first 100 or so you see are fraud clinics. There are people who do not have scientifically verified cells that have claims they can cure every disease. A lot of anecdotal stories I can tell you, they're horrifying. If you are contemplating a stem cell therapy for you or yours, you have 2 questions to ask to know if it's real or fake. The first one is you ask the people who are going to charge you for this therapy, give me the name of the person who is the head of the institutional review board that oversaw the clinical trial when you first started this out. Because that person knows whether it was safe or not whether it's effective, just safe. The second question is, give me the name of the FDA protocol that showed it was not only safe but effective. If they can't answer both questions directly and rapidly, you should suspect that they're going to take you for your money and give you nothing in return.
Yes, is there any hope for heart improvement with stem cells?
We have, and many others have, isolated from embryonic stem cells or iPS cells, the cells that participate in forming heart muscle, and other cells that participate in forming the blood vessels, and others, a third one, that makes the smooth muscles that keep the arterial pressure up. Depending on which cells were killed during your heart attack, you probably need the muscle-forming ones most of all. There are lots and lots of labs doing it. There are early clinical trials, and as you might have expected, the first group put blood-forming stem cells in. We had to publish in a journal. That was a fraud. The people never retracted their paper. Then the others keep showing mesenchymal stem cells. They didn't have mesenchymal stem cells. All it does is form scar. We still have to get into real, serious clinical trials with large animals, similar to humans, that have the lesion. Joe Wu at Stanford is doing it. Reza Ardahalli at UCLA is doing it. There are groups trying to do it. And they're serious. Charles Murray in University of Washington. So there's hope. And of course, if we still can do it, if we're funded to do it, if there's no political criminal activity by doing it, which is a major problem right now. Many states don't allow it. We had to stop a clinical trial with our human brain stem cells in Ohio because they had a ban on it. But if we get there, it may be 5 years, 10 years, it may be 50 years. It may be your kids, not you, who get it. But if we don't start now, we're not going to get there. We're just always going to be saying in ignorance, well, let's try this, let's try that.
Is there any work being done in treating ALS with stem cell research?
Yeah, there's, I believe it's a group at UC San Diego that's developing human neural stem cells. Also, Cedars in LA, I can't remember if it's Cedars. And there are many places. There's a place in Israel, Jerusalem. So there's a number of people who are going one step at a time to see. Now, ALS is a difficult disease because you lose the stimulus from the motor cell, not a stem cell, but a motor neuron, to stimulate the right muscles to react appropriately. Stem cells that are brain stem cells, we don't know how to make them not only make that cell efficiently, but to go to the place where it normally was and make the same connection so that when you think about moving your thumb, you actually move your thumb. And of course, all the way down the track, you have to have an insulator. So it's electrical, not chemical, so it goes fast, if you're thinking to move your thumb. So it's complicated. But I believe that step by step, we'll find out when we finally get to clinical trials if it works. Now, the iPS cells that you asked about, I was the head of the National Academy of Science Committee, or panel, on making iPS cells on nuclear transfer to make pluripotent stem cells. And I said back then, and say now, the biggest use of it will be to take somebody who has genetic ALS or genetic Parkinson's or genetic Huntington's or any of the genetic diseases, take their skin cell, make a pluripotent stem cell, and now make the motor neuron, the oligodendrocyte that insulates it, the astrocyte that feeds it, the motor end plate on the muscle, and say, okay, where is this disease happening? So it opens up a rationale to get there. But it's still 10 to 20 years, I predict, before you start seeing the cures. But 100 years from now, 1,000 years from now, it should be better.
So, sorry.
I have a question actually regarding the patients in the trials, since I know you've done some human trials. I was wondering about the ethical conversations you've had about doing the true stem cell therapy versus the ones with others, because after you've done a few of them and you've seen quantitative, like you can actually show that there's a benefit, do you stop giving the other treatment because it's not as effective anymore, or you have another treatment that actually shows promise? And so I know you're supposed to, when something's not working or when something is absolutely working.
Our problem in that breast cancer study, it was a real stem cell trial, wasn't ethics unless you talk about the ethics of making money. We didn't have an ethical problem. When we first figured out a system that would allow us to isolate these cells, we had to use leftover human fetal remains from a woman's choice abortion.
Right.
And we had to work out the ethics before we ever made the first one.
I meant after the trial was started. So you have something and you're showing that it's proven to work, and now you have all of these women who are dying because they didn't get that particular therapy, and you're proven that your other therapy does actually work more successfully. And
well, I'm happy if any therapy works more successfully. I'm trying to get the ones that we study and understand to work successfully. And as I think I pointed out, in every case, we got to clinical success and they aren't therapies. So the locus of that problem is clear now. It's not us. It's not the agency now in California that funds us. It's something else.
so for what it's worth, I actually have embryos that my husband and I may or may not be using at some point, and we're donating them to research afterwards. It's just, this is important to us.
So say it again?
My husband and I have embryos in case we decide to if we have difficulty getting pregnant, but when it seems like we're not going to have that issue, we're donating them to therapy because
Great.
It's important.
It's very important. There are states in which you can't do it.
I know.
I was wondering if you could comment on the increasing practice of cord blood banking, private cord blood banking. A lot of young couples are paying a lot of money for it, but there is no real regulation of the private cord blood banking industry.
Yeah, I actually have been heavily involved in this issue. So the cord blood of a child has the same genetics as the child. So given, if you don't modify the cord blood, the kid gets leukemia, the kid gets a degenerative disease of the blood. That cell won't fix it, because it's got the same problem. So you have to genetically modify it. Now, if you go to the ads of the private cord blood banks, they say you would be lax as a parent if you didn't pay your $2,500 plus $250 a year to have that kid's cord blood. Cord blood availability has been there for a long time, and the New York Blood Center is one of those great centers that have done it since the 70s. So I called up the head of that center about 10 or 12 years ago. I said, you have over 100,000 samples, and you have the freedom to use it on the patients if they request it. How many have used it? Carlos Irving. He said 1, and it was inappropriate. Now, on the other hand, if they were public cord bloods, then you could find, and people do find, who might be a match at the major genetic locus that allows transplantation immunosuppression to succeed. And many lives have been saved because of that effort. So, the private cord blood banks that charge you like that are using very clever misinformation. They say you would be lax if you didn't save those stem cells from your kid for your kid. And here's the diseases that stem cells will fix. Their whole list are genetic diseases. So it won't fix. It's It's the same thing I've been talking about. It's money.
Thank you for your presentation. We learned in one of your slides that if a transplant of an organ is done, along with purified stem cells from the donor, that organ will not be rejected. What is the effect on the other organs in the target patient?
No. Because what you've done, you've used antibodies, at least in our situation, to wipe out the immune system and the stem cells, not the other organs. You rapidly replaced a week later with healthy stem cells from the donor. That day or a year later, you could put the organ in, but within a week or 2 weeks of the transplant, there's nothing going on. Now, if you had co-transplanted T cells, there would be an immune reaction against the host going on. Right? That you don't want, we don't want, but that's what every, quote, stem cell therapy does, is they include T cells. And then they spend all their time trying to dampen the reaction of those donor T cells reacting against the host.
Thank you.
Okay, we have time for one more question. Who wants to do that? That was the first hand.
This is the last question.
The last question.
This is the last one.
Yes, you had multiple sclerosis on your list of diseases. As I understand it, there are a number, like over 100 genes that are associated with multiple sclerosis. How are your techniques going to narrow down, or is that not really an issue?
Even though there are many genes that are involved as participating or possibly participating in that disease, the final common pathway is you have T cells in your own body that are attacking the oligodendrocytes in your brain and spinal cord. That's the common path. We can wipe out the T cells. We can replace with a predefined genetic donor stem cell that doesn't have the predilection and hope that it works. Now, me saying that means we're at the level of mice today. So it'll take time before you get to humans. But again, as I said before, if you don't start, you won't get there.
Okay, thank you very much, Irving, for the presentation.